Okay, I'm going to give you really more of a fly 10,000 feet above the surface view of what my lab is interested in. We started off a decade ago trying to understand the question of interneuron diversity, thinking it was going to be a developmental genetic problem, and increasingly realizing that it's kind of a two-step shuffle between an early genetic program and a later program that's driven by local signals and activity. So I'm just going to honestly give you an overview and a flavor for what we're interested in, and maybe take your hand and go along on a journey of how we've kind of figured out at least some things about how interneuron diversity is made. So can I guess I can do this, right? Okay, so this is the kind of picture that Ramon y Cajal and Golgi saw when they looked at the brain. And exciting as it is, it's a little hard to understand. And so the first step in trying to understand it really was to sort out the various cells in this mesh of cells and understand what is the diversity. Yes? Ah, okay, got it, right. I'm not used to this. Usually maybe I can just stick it in my pocket there. Okay. Does that work? That works? Okay, good. So we were just trying to figure out the diversity of the cells, and what they saw, as we know, is that about 80% of the cells in the cortex are the so-called principal cells. These are the conduits of information to kind of take an external world to represent it internally. And over here we have the interneurons, which look a lot more diverse, but in fact they only represent somewhere around 10 to 20% of all the cells in your brain, probably a few more in humans than in lower species. And these take these internal representations, bind them together, and allow you to really put action on what is otherwise just static sensory information. So really understanding how interneurons function at its basic level means understanding how the brain is wired. And the neocortex is actually a wonderful place to study in terms of sensory perception because it has canonical pathways that are iterated irrespective of whether you're looking at the visual, the somatosensory, or the prefrontal cortex. The size of the layers vary a little, but the basic pattern and logic of information is maintained. So sensory information generally comes in and goes to layer 4. That's transferred up to layer 2, 3, where it starts getting associated with other areas of the brain, ultimately down back to 2, 3, and then to layer 5, and then it leaves the cortex. And as you'll probably see with some of the later talks, that's a gross simplification of how it works, but it is a fairly stereotyped flow of information that we know is conserved. And the information doesn't just simply pass through the cortex, it needs to be modulated, coordinated, and amplified. And what seems to do a lot of that are the interneurons. So they can shunt input, shunt output, shunt throughput, and perhaps most interestingly, there we go, it can coordinate the timing of how cells fire. So the interneurons are inhibitory. They don't create information. They can simply modulate the timing and the throughput of information that's already in the brain. And that really gives them some really impressive power in terms of allowing the brain to function. And one of the most obvious manifestations of brain activity are the oscillations we see in brains. So long before we understood them, and that's to imply we still don't understand them, we knew that the brain has a series of characteristic oscillations. So if you do an LFP or even a surface recording, you can see these characteristic patterns of activity ranging from the very slow delta up to the very fast ripple up around 200 hertz. And if you look at these oscillations, what a variety of people, probably most prominently Peter Somogyi and Klausberger, who really studied this in the CA1 part of hippocampus, is that they found that the interneurons are entrained by these oscillations. So here you see the basket and the axonic cells fire at the peak of the theta in CA1, and the bistratified and OLM cells fire at the trough. So this generalized activity of the brain actually selects in the duty cycle when these interneurons fire. So what are these oscillations? Well, as I said, we don't understand them, but we do know that they correlate with emergent behaviors, foraging, sleep, sex, all the things that make life fun. And it turns out that not only do the interneurons get entrained by these rhythms, they can in fact generate them. So beautiful work by Chris Moore and Jessica Carden showed that if they stimulated basket cells at a requisite frequency, they could actually induce gamma in the brain, which is certainly thought to be important and disrupted in things like schizophrenia. So that's kind of the yin and the yang of the interneurons. They're both elemental to control the flow of information in our brains, and probably tightly coordinated, if not causal, in some of the conscious thought that we do. So I was told this is supposed to be interactive, so I'll pause awkwardly every few minutes, just stand here and do nothing until someone asks a question. Yeah, it's going to get uncomfortable. Okay. So, Ramon y Cajal, there's no neuroscience symposium, talk, or even conversation is allowed to occur without invoking either Ramon y Cajal or Charles Sherrington, right? So I'm using Cajal. And you know, this is a statement that Cajal made back in 1923. It's a little verbose. The functional superiority of the human brain is intimately linked up with the prodigious abundance and unaccustomed wealth of forms of the so-called neurons with short axons. What does that mean? Well, at least in part, if you want to simplify it, really he was talking about the interneurons. So Cajal, who was certainly the master of morphology, looked at brains of everything from rabbits to squirrels to dogs to humans. And you know, an obvious question was, what made us special? You know, why can we talk, build airplanes, and dogs basically just act happy? And he concluded that the interneurons were somehow tied to that. And the thing that really attracted him to it was this incredible diversity of the interneurons. And although this was entirely based on morphology, he correctly inferred that they also were functionally diverse. Now, a point I'd like to make here is that he did not actually understand that they were inhibitory. In fact, him and his most famous student, Laurenti Deneau, did think that they were important in relaying information, but they actually thought they were excitatory. And it actually took some 50 years till Chuck Rebeck actually identified that these cells were all carrying the neurotransmitter GABA and were inhibitory. So the question I'm interested in is, you know, how do we create these wonderful different flavors? And you know, every field needs its controversy, and ours is the lumpers versus the splitters, okay? So the lumpers, and I'm probably a slightly reforming lumper, says there aren't that many types. You can choose a column, and then based on morphology, where they actually connect to other cells, so do they give input to dendrites, soma, axons? Are they bulk transport? Are they targeting other interneurons and hence are disinhibitory? What kind of markers they have? Do they turn on parvalbumin, somatostatin, VIP? Or probably at least the best correlative function, how do they fire and how do they actually depolarize when you put some current in them and get them to threshold? And the other school of thought is the splitters. And they kind of posit that you treat this like some sort of menu item in a deli and choose one from each column. So if you take a basket cell, parasomal input, parvalbumin, and fast spiking, you have one of the types. And an enormous amount of energy, at least from developmental people, goes into this argument about how many types of interneurons we have. And I'm going to take the radical position that this is a massively stupid argument. And the reason it's massively stupid is because God did not look down on the sixth day and said, let there be interneurons, and it was good. There's nothing in biology or evolution that says a cell type as a distinct entity like your friend George has to exist. And so, we spend all this time trying to say how many interneuron types there are, and no one's done this better than Peter Somogyi, and he now has, I believe, 26 different types of interneurons in CA1 alone. And I'm not mocking that work. That work is beautiful and elegant and is wonderful if you happen to be studying in CA1, but if you move over to cerebral cortex or the striatum or the amygdala or, God forbid, the diencephalon, they all have interneurons in them. And all those interneurons have a lot of similar properties, at least within the telencephalon. And as I'll show you, the question is that these cells don't get a distinctive, unique fate like that. It doesn't just appear. It's progressively imposed on them, first by a genetic program and then later by local cues where they go. And what I would say is the more profitable question is not how many types, but if you look at any given emergent property, anything that you could do principal component analysis, like these 4 parameters, and ask where does that information come from, I think that's the useful way to go. And so what my lab's been trying to understand is not so much how many types, and though in fairness there seems to be discontinuities, so if you actually took all those components and mapped it in n-dimensional space, you would find clusters suggesting that, at least based on a quantitative point of view, there are individual types. Many of them exist in a kind of a spectrum from one type to another. And so I still think that the thing we really need to challenge ourselves is to understand where that information comes from, how is it imprinted on the cell. And you're going to hear Paula Arletta talk a little bit later, and she's done some absolutely elegant work looking at some of the transcriptional programs. And I think she would, well, perhaps agree with me. She may shake her head sadly when she comes up here. But that those pieces of information are the result of gene expression. That gene expression is the result of largely environmental cues, and be them early or late, that tell the cell what cell it needs to turn on and when. So this all started with my good friend John Parnavalis, a humble Greek man. And back in the 1960s, Angevin and Sidman were really interested in understanding the inside-out generation of the cortex. As a matter of fact, they discovered it. And what they realized is that if they used tritiated thymidine, a really groundbreaking tool at the time, that you can give a pulse and birth date cells, what you find is that the cells are born in an inside-out fashion, and that the early-born cells go deep and the more superficial cells go superficial. The later-born cells go superficially in the cortex. And what John did was try looking at it for the interneurons. And what he found was that the birth dates for interneurons didn't bear a hell of a lot of resemblance to the birth dates for the pyramidal cells. And he actually realized back then, which took the rest of us about 30 years to realize, that this implied they had a different and separate origin. So my entry into the field came with the work of John Rubinstein. And John was really the pioneer in looking for genetic markers that differentiated the early brain in different areas. So this is a kind of an explanted lateral part of the cerebral hemisphere. This is a coronal section. And the brown area there is a gene called DLX. And what John recognized is that this was one of the early, and as it turns out, fundamental genes that specifies the subpallium, the areas where all the interneurons turn out to come from. And this progression actually made for a huge jump in logic. So what you see if you look at E13.5 in a mouse, or maybe 3 weeks postnatal in a human, is all the DLX is confined to ventral areas. But if you wait a day or two later, these guys spring forth and migrate all over the place. And you might have just thought it was changed gene expression, but Stuart Anderson and John Rubinstein, through an elegant experiment where they cut off, there, thanks for doing the dissection for me, cut off the subpallium from the pallium, they realized that you could deplete all the interneurons dorsally by breaking the land bridge between ventral and dorsal. Now, I thought this was one of the great moments in neuroscience. I asked John for a picture to show this. and that's the one he gave me. It didn't really get the somber, you know, kind of elevated feeling I was looking for. It's also confusing because there are 4 people in the picture, so who's John? So I decided I better write labels on there, and there's John and Stuart, regaling in their discovery that interneurons are subpallially derived. So this is really kind of through work of many, many people, including Oscar Marin, Nicoletta Tequi-Caceres, Nenad Sestin. Many, many people have contributed to this. We've realized that all the interneurons in your cortex, and actually, as it turns out, striatum, hippocampus, and amygdala, come from one of 2 structures. So if we took an embryonic brain in a human at about a month, or in a mouse at maybe 12 days of gestation, and we just cracked open the dorsal part of the cortex and peered down into the lateral ventricle, what we would see is that there are these 3 bulges going into the lateral ventricle, the medial, the lateral, and the caudoganglionic eminence. And we, some years ago, as well as Stuart Anderson, started trying to understand what cells come out of these various eminences. And the approach we took was to take a genetically marked donor animal, where EGFP was in all the cells, dissociate the cells, and then inject them in a host. We did this using, and we'll see if this works, there we go, ultrasound backscatter microscopy, and that allows you to take a needle and inject in and put the cells back in. This is fate mapping like you might do in a frog or a chicken, but we did it in mouse. And when we did that, it became very quickly clear that different part of these eminences made different cell types. So the lateral ganglionic eminence mainly made medium spiny striatal cells, as well as, yes, a question, good.
Sorry, probably you mentioned it before, but I get lost. At what time did you do this?
So this was done before they're born. I actually skipped that. Embryonic, so we did the transplants anywhere from about E10 to as late as E14. We did a few different time points. Took them out, dissociated them, and put them chronically, homotopically back in. So we stuck them right back where they came from, and this is the result. It turns out if you put them in the wrong place, they still do the right thing.
And you check it with postnatal day or right day?
Yeah, so we let them survive. They actually, the animals aren't damaged by this procedure, and when they're born postnatally, we could then look at where the labeled cells ended up. And that's what you're looking at down here. This is about P21. So this is 3 weeks postnatally. So this is done at E12, and that's P21. So there's 3 weeks of time that have elapsed between these 2.
Did you check in the adulthood?
That is pretty much adulthood there. Okay, we did it at 3 months too. Does that make you happier? Yeah, it doesn't change a lot after this. Okay, this is really reflecting where they end up in the adult. And so what you see is the LGE mainly gave medium spiny cells and interneurons in the olfactory bulb. The CG and MG gave rise to all sorts of ventral areas, which are interesting, but not really my topic today. But what was obvious was that if you're just looking at interneurons, it really was the CG and MG that made the interneurons in the brain. And that really, this I think made 2 points, one of which that the interneurons come from these ventral areas, and 2, that different types come from different areas. So this was done in collaboration with Dan Turnbull, a very good and old friend of mine who invented the ultrasound backscatter microscope for this sort of thing, and Arturo Alvarez Buya, who was at Rockefeller then but has gone on to UCSF. And he actually came up with the methodology that allowed us to do these nanoliter-sized transplants. So what we found as we moved along was that interneurons, although there are many different types, can be divided into 3 basic groups. Those that come from the MGE, which are parvalbumin and somatostatin, and those that come from the CGE, which uniformly express this ionotropic serotonin receptor, the 5-HT3AR. And what it doesn't account for, of course, is that there are a whole lot of different interneurons that have 5-HT3AR. There's at least 2 types, basket and chandelier, and probably other types in here. And as my colleague and friend Bernardo Rudy has shown, there are actually an amazingly diverse number of somatostatin interneurons. So the success was dividing it into 3 non-overlapping categories. The failure was that there are many subcategories we couldn't account for. And I handed the tools, the genetic tools we used for this, over to Chris McBain, who's a friend of mine who works at the NIH and has done superb work on the hippocampus. He actually discovered the OLM cell some years ago. And we expected it to be exactly the same thing. And so one of the things that is very characteristic in our analysis is that all the Reelin cells come from the 5-HT3A population. But when he looked in the hippocampus, somewhat to our surprise, about a half to a third of them came from the MGE. So this seemed to break the rules. And then when he looked at somatostatin, a cell type which entirely comes from the MG and the cortex, when he looked in the hippocampus, again, a bunch of them came from the CGE. So the rules of site of origin to type, which we thought could be predicted entirely based on site of origin, turned out to be a little more complex. And there's two ways you could explain this. One is that the MGE has little dedicated areas that go to the cortex, another part that goes to the hippocampus, another part that goes to the amygdala. Or you could posit that there's learning on the job, that basically a common pool comes out of one of these structures, and that's something that happens after they migrate, customize them where they go. And I'm not going to actually show the data that convinces me it's the latter, which is lineage data, which essentially shows that a common lineage can give rise to interneurons in cortex and hippocampus, cortex and striatum, even cortex and globus pallidus. And Corey Harwell, who used to be with Arnold Krickstein's lab, did the exact same experiments and came to the same result. And so this makes me believe that what you do is you have a common pool of interneurons, and there's Chris McBain, because we always need a Scottishman. A common pool of interneurons that populate these various areas. So this is a map. This is just showing excitatory versus inhibitory cells. But if you concentrate on this side, the blue and the yellow, in a way that's supposed to be quantitative, shows you the percentage of interneurons in the hippocampus, the cortex, the medial and lateral amygdala, the striatum, and the entorhinal cortex that come from the MG versus CG. And you can see that many, although not all, derived interneurons from both those places. And what I just told you is not only do they derive interneurons from those places, but a common lineage can actually populate one of its daughter cells to the cortex and another down to the striatum. So how do we do this? How do we make these diverse interneurons? So I've put out there a model. I emphasize it's just a model. It needs to be tested. And that suggests that interneurons are made in what I call a two-step shuffle. The early step is what I call cardinal specification. And cardinal specification is effectively what information is imposed on the interneurons based on where and when they're born. So essentially, as soon as they become postmitotic, I believe they're cardinally specified. I believe the diversity is actually surprisingly finite. And I've gone on record as saying, and I'm being recorded, so I'm going on record again, as saying that there are maybe as few as 10 different interneuron cardinal types. And so that flies at the face of the at least 10 times, if not 100 times more of interneurons that you can find in all these areas. And so how does that occur? So cardinal specification, I think, basically lays down things like intrinsic physiological properties. When you're born, you know you're a fast spiker or a rebound, a burster, or a delayed spiker, or a stutterer, all the great little physiological terms that describe how they fire when you depolarize them. Things like parvalbumin, somatostatin, and 5-HT3A, these, it's not clear exactly what they're needed to do functionally, but they seem to be laid down very early and are immutable. They're targeting. So by targeting, I don't mean specifically what cells connect afferent and efferent to them. I mean where they target to. So, basket cells are called basket cells because they form basket around the recipient. You want another question? Because you're the only one asking. No one? Come on, guys, you're killing me. Okay, so basket cells are the only ones, are called basket cells because they form basket around the targets they target. Chandelier cells make axoaxonic projections, and cells like VIP are disinhibitory, so they target inhibitory cells. Somatostatin target the distal dendrites of pyramidal cells. So each of those specific ways they innervate other cells and whether they innervate excitatory and inhibitory cells, I think is an intrinsic property that they already have when they're born. Now, by contrast, I've come up with another term I call definitive specification, which is their circuit integration. And so although there are real strong similarities between the OLM cell in the hippocampus, the Martinotti cell in the striatum, and the low threshold spiking LTS cell in the striatum, they clearly have completely different environments, and as any anatomist can tell you, they connect up completely differently. But because I believe they have a common origin, we believe that actually the information that tells them how to wire up is only determined after migration. So that's learning on the job. And part of that is, of course, their axonal and dendritic morphology, which are very unique to different layers. And again, obviously that determines a lot of your afferent and efferent connectivity. And so that, again, is learned on the job. So early patterning starts off with extrinsic signaling. This is actually kind of near and dear to my lab's heart. We worked on it for about a decade. And things like FGF and sonic hedgehog and retinoic acids basically create local cues that program progenitors in order to tell them generally what sort of transcriptional code they should turn on. And that results in structures forming with different transcriptional cascades, like these two structures, the medial and caudal ganglionic eminence. And that results in a very complex genetic set of genes that are turned on in each one of these. And we know some of the players, we don't know all of them. But I think the first challenge in understanding interneurons is really understanding how different are the genetic programs in the MG-derived progenitors versus the CG-derived progenitors in order that they can make the cardinal types that are ultimately going to populate the brain. And so this complex genetic cascade is started by a gene in the MG called NKX 2.1. About 15 years ago, Laurie Sussel with John Rubinstein showed that if you do a knockout for NKX 2.1, the MG respecifies to what they said was an LG, but could also be a CG. In other words, the whole structure changes its fate. In a more specific removal of the gene that we did about 10 years ago, we created a trick where we could take the cells, have them have NKX on till they're born, and just as they were just being born, we used a genetic trick to remove NKX 2.1. And we got what I called was class switching. So a cell that should have taken on either a Martinotti or a basket cell fate as the most likely outcomes totally changed its program and took on, for what all the world looked like, and by that I mean by its shape, its connectivity, its firing, a CG identity. So we posited that NKX is a master regulator determining MG versus CG fate. But downstream of NKX is a whole series of genes that, yes, question two. Yeah, slightly different. CGE cells tend to target more superficial areas of the cortex. They're more delayed in when they get there. But, and there's certain areas CGE cells don't seem to go, like the striatum or the amygdala. And to be honest, we didn't look carefully enough to know whether all that was true, though I suspect it is. But we mainly looked in the cortex. And so they went more or less to the same areas, but they shifted in accordance with their fate from being mainly in the deep layers to mainly in the superficial layers. Question 3, anyone? No, okay. Okay, so Vasilis Paknis and Stuart Anderson have looked at this gene, LHX6. That's effectively a direct target of this. You need NKX to turn on LHX6. But NKX disappears and LHX6 persists. And LHX6 seems to be not a gene that tells you to be MG versus CG, but really a gene that you need in order to make the emergent properties of all the cells from the MG. So if you have an LHX6 mutant, neither the basket cells nor the Martinotti cells are totally normal. A gene that Jeff Macalis and my lab both worked on and came to similar conclusions on is SOX6, which seems to be a further and later differentiation gene. It's actually genetically downstream of LHX6, and it's one step along. And then you get a split between this gene SAPB1 that Vasilis and I both studied, which seems to be about as close to a master regulator of Martinotti cells as you could ask for. Really cool gene. It's a transcription factor. It's a histone modifier. It's 2 transcription factor modifiers in one. Very exciting gene. And it is very important for the Martinotti cells, but also for the basket cells. And another gene we've discovered, we haven't yet published on, is a gene MEF2C that we know is activity regulated from the work of Mike Greenberg. And we know that if you knock it out, the basket cells just die. They're born, they migrate normally, they get to the cortex, and then pretty much a large proportion of them die. So, and then downstream right here, we have a gene we've published on last year, catnap2, which is from the neurexin-like family, and it's needed for presynaptic organization of basket cells, and then some calcium channels, the CACNB4 and CACNA, which are calcium channels of the PQ family, which are needed for the fast release of basket cells. So what you can see is there's a whole series of genes that come on at different times and allow the cell to realize its adult fate. And what is interesting here is, well, these are really clearly intrinsically driven. SOX6 is only on in the cortex, so that's probably turned on later. SAPB1 is actually activity regulated, as MEF2C is, and so that's clearly requires some cues post-migration. And these genes are mainly on, well, this one's on in interneurons everywhere, but this one's mainly on in interneurons in the cortex. So what you can see is that this cascade is not only sequential, but probably requires different bits of information to be realized. This is really evidence that we've accumulated over the last 6 or 7 years as we've tried looking at that later step. So we already knew that interneuron classes were specified in a gross sense by genetic programs, but we really wondered, what did the later step? I keep emphasizing that they learn and adapt on the job. I've shown you a few genes that are activated by activity. And so the question was, what is that role of activity in telling the interneurons what to do? And this is a slide from Rosa Kozart, and it basically shows that your cortex doesn't turn on like booting up a computer. It basically has an initialization phase, and the initialization phase is started by intrinsic activity that's spread probably mainly through gap junctions in the forms of slow waves in the sub-1 hertz, SPAs, spontaneous plateau amplitudes, and these are all gap junction related. Eventually you get glutamate driven activity, which may be from the primary neurogenesis of subplate and Coleretzius cells, and they form what are called early network oscillations. And then just before they kick online and GABA is still depolarizing, you get what's called giant depolarizing potentials, which are informative in terms of making the cells start firing in a slightly more sophisticated, less coordinated fashion. And what we noticed was that if you look at the various stages of interneuron development, from proliferation to radial migration to morphogenesis and synaptogenesis, really in this phase here is when you go from the SPA to CNO and GDP phase. And so the question was, does those early activities have a role in instructing the interneurons and how they should develop? And properties like this, which is the switch from GABA being depolarizing to hyperpolarizing as a result of KCC2 expression, which is a potassium chloride exchanger that basically pumps a whole lot of chloride out of the cell, changing the effect of opening a chloride channel from being depolarizing to hyperpolarizing. So the cells change in how they respond to GABA at a critical time. And so the idea was, is GABA, for instance, one of those signals, that switch, that tells the cells what to do? And in fact, work from Frank Palou has actually shown that this switch is probably really important for telling the cells when to stop migrating. So GABA is depolarizing in the interneurons until they get to the cortex, and as they integrate in, GABA becomes hyperpolarizing and they stop migrating. So that switch is basically taking the same signal and by interpreting it differently, telling the cells something different they should do. Okay, so what we did as a first pass of this is we used an inward rectifying potassium channel, which basically gives this huge leak current of potassium. The reversal potential for potassium is about -90, And so when you overexpress this in cells, it basically makes them deaf to signals. So they still get their normal excitation, but because their resting potential has been dropped somewhere between 10 to 15 millivolts, it can't excite the cells the way they would normally. And when we did this in CGE cells, we noticed that some of them migrated to layer 4 instead of layer 2, 3. And that was specifically the Reelin population, which is our code for neurogliaform cells, whereas the VIP population didn't seem to care that much. So activity seemed to have a real effect on these Reelin expressing neurogliaform cells. Yeah?
Did they change the way they spike?
They did not. Their intrinsic properties were the same. So they were in the wrong layer, and as I'll show you in a minute, their morphology was really affected. Sorry. Their morphology was affected. So instead of being beautiful neurogliaform cells, they were these little stumpy neurogliaform cells that didn't look too normal. Though surprisingly, you're right, the intrinsic properties were the same. The markers were more or less the same, though Reelin turns down a little. And just based on the iPSCs and EPSCs, they seem to actually wire in. But you'll see, actually, they wire in differently. So this is going to take a whole lot of experiments and reduce it down to one slide. So what I just showed you is that when you block activity, the cell is not only in the wrong layer, it morphologically doesn't develop normally. So we wondered, who's giving that activity, right? So if activity is going to it, you got to know who's providing it. And Ed Callaway, and in collaboration with Martin Goulding, who made a genetic method, came up with a rabies method where you can look at the afferents. I think Benedikt's going to talk about this in his talk. And so what you do is there's a whole lot of tricks involved in this. I'm going to let Benedikt tell you about them. But basically, you inject rabies virus, and that infects your starter cell. And then it transfers the rabies to all the afferents. And what you see if you do it with a neurogliaform cell is it gets a whole lot of cells from the cortex and subplate, and surprisingly, a huge number of inputs from the thalamus. So work on the neurogliaform cell in the adult suggests it doesn't have such a strong thalamic input. So this seems to be an unusual developmental feature, that they get strong but transient thalamic input to the neurogliaform cell. Work I'm not going to tell you about on the Martinotti cells shows that that's true of them as well. So at least these two classes of interneurons have this unusual developmental step where they get afferents that are transient that disappear and remodel at a later stage. So what happens if you block the cortical input? Does that have any effect on the cell? And in fact, not much. They didn't seem to care. So if you put tetanus toxin and block out the entire cerebral cortex with it and look at the neurogliaform cell, morphologically it looks pretty normal, right? And so then we put tetanus toxin in the thalamus using a genetic trick, and that gave us the same phenotype we saw when we blocked activity intrinsically using the Cur 2.1. So that suggested that the thalamus had some privileged position in terms of the morphological development of these cells that the cortex didn't. And that led to the obvious question of what's different between the two. They're both giving glutamatergic inputs. How is one allowing them to morphologically develop and the other not? So we did some physiology. We put channel rhodopsin in the cells of the cortex and then stimulated the interneurons, put on light and looked at how they fired. And we got, what you might expect fairly typically for a glutamatergic input, an NMDA and an AMPA response. And then we did the same thing on the thalamus. And again, we got it, slightly more pronounced NMDA currents, a little higher, and probably, if you integrate under the curve, a little bigger. So they both get AMPA and NMDA. Their ratios are a little more biased for the thalamic ones than the ones in the cortex, and it suggests that these may actually go to different receptors on the two. That was actually our guess. So in order to investigate that, we wondered if the calcium permeability of the receptors they were on were any different. And so there's a drug called infempridol that blocks the calcium permeable NMDA receptor. And when we did that, we got a response on both, but the response was actually quite a bit more pronounced on the ones from the thalamus. So what we think from this is that these neurogliaform cells are getting inputs from the cortex and inputs from the neurogliaform, from the thalamus, but they're actually going to different receptors on the cell, and those receptors have different calcium permeability. So that the excitation from the thalamus is causing a bigger calcium influx to these cells than the inputs from the cortex onto the same cell type. And I think this is important because I think that is probably driving transcriptional programs, and certainly seems to be accounting for the morphological defect you see when you block the thalamic inputs. So here's kind of a summary of what we think. Thalamic activity is impinging on the cell, activating this and causing calcium influx. That's creating a calcium signaling cascade, which we know involves calcineurin, and we have some work, which I haven't published, which looks at the role of that, and that leads to the differentiation of the cell. So if you block the NR1, you affect the cell. We did genetically block the NR2, same phenotype. And in fact, if you block calcineurin, you also get the same phenotype. So this kind of forms a nice story where you imagine here you have a neurogliaform cell freshly arrived into the cortex. The thalamus gives its input, and that gives this cascade of activation of NMDA, calcium influx, calcineurin activity, and a happy, well-differentiated neurogliaform cell. So what was really cool, and this actually has got me really excited for this cell, and in fact, work that Li-Chin Lo has done on pyramidal cells, but he's talked about, so I feel it's okay to talk about it, but hasn't published, is that when he did this exact same manipulation to pyramidal cells, it didn't seem to have much effect on them. But when we did it to the neurogliaform cell, it had this morphological effect, but then we teamed it up. So we removed the NR1 channel, so we got rid of signaling through this NMDA channel, and then we did rabies tracing. And here's what we got. So I'm going to toggle this a few times so you can see it. So this is the normal connectivity you get in the cell. About 2/3 of it's coming from the thalamus and 1/3 from the cortex. And that's what happened when we removed the NR1. The thalamic input drops to about 10% of what it should, but it's made up for. The cortical cells now make more connectivity. And so I think this is a really cool observation, because what it implies is that the early connectivity is not only driving the morphological development of these cells, but specific afferents are instructive in telling the cell how they should wire up. And that when you remove the thalamic input, you not only disrupt its developmental program, you actually fundamentally change the connectivity of the cell. So now it's getting way more cortical input and less thalamic input. So you'll hear more from the later speakers about cortical networks, but from a developmental point of view, the cortex is trying to reconcile two types of inputs: ascending sensory information, which people like Heiko Luhmann and Rostam Korsapov have shown seems to be driven by the periphery, and recurrent cortical activity, which is the actual processing. And that balance is really critical to the way our brains function and process the external world. And what this implies to me, and I think there's a lot more to learn, is that these interneurons help integrate that and help control that flow. And in fact, when you disrupt it and mess with it, you actually change the basic connectivity of ascending versus recurrent information to these cells. And I suspect probably fundamentally reshape the way the cortex processes information. So I think that's pretty, so these are the terrific people who've done it. Natalia DeMarco has her own lab at Cornell now. Theo Karianis, who will be joining the University of Zurich shortly. Rashid Priya, a graduate student who's almost done, going on to Arturo Alvarez Buia's lab. And Shebnam Tunç Demir, who's joining Renee Han. So this is my reminder that, you know, I go up and give talks and say highfalutin things, but they're the ones who really get the work done and really deserve the credit. So at that, this is New York in a slightly blue haze. These are the many, many people in my lab, and here are some of the people who have been really critical to some of the work I talked about today, and we'll take the rest of the time to take questions. Thank you. Yeah.
Have you looked at any behavior phenotypes of disrupting the development of the neurogliaform cells?
Yeah, so that would be wonderful to do. In this kind of, the way I'm showing you will never give you a behavioral phenotype, because in order to make it cell autonomous, we do it very, very sparsely. So we're only affecting a small, small fraction of the neurogliaform cells, too small, at least in my estimation, to give any strong behavioral outcome. So using these methods, it's really good to figure out what's happening in a cell autonomous basis. It's absolutely crap for understanding at a behavioral level. So there are a number of genetic manipulations we've done where we can remove genes like NKX2.1 or another gene we've worked on, PROX1, which is the CG-specific gene. And these have broad and ubiquitous effects on the interneurons that come from these areas, though not entirely specific to just one type of interneuron or even interneurons on their own. And those have behavioral consequences. So if you mess up MGE interneurons, PV and somatostatin, individually or together, the most obvious behavioral outcome is you get severe epilepsy, which kind of makes sense. Though I was a little surprised. I wasn't sure the Martinotti would be sufficient, but I have to say at this point I'd say that Martinotti cell effects are sufficient to give you epilepsies. That in part probably reflects another story which I haven't talked about, which shows that the Martinotti cell actually is needed for the development of the PV cell. And that when you screw up Martinotti cells early, it actually affects the way the PV cell develops later. So that's work that Shebnam has done, and we hope to have out in Neuron fairly shortly. The CGE1s, PROX1 certainly affects CGE1s in a pretty hard-hit way. And the animals, if you just kind of stare above the cage and poke them, they don't look so bad. But that's not a very sophisticated behavioral analysis. And I strongly suspect that as we get better at it, we'll see more. And in fact, there is one manipulation, which again is not terribly specific, when you mess up cholinergic input, where you get repetitive stereopathies to the animals, which kind of implies that they may have something to do with breaking stereotype behavior, and if you want to stretch it, may be the kind of defect you'd expect in something like autism. But there's a lot more to do on that. It's a great question.
So once those interneurons are in place, what guides then the typical connectivity they do in cortical microcircuits? Is this the same genetic predeterminants, or is it more interaction between the cell types that are already installed?
I think the answer is probably most honestly yes, which is both. So the way I see it, I have certain stories. So once you're a PI, you realize that you constantly tell people stories and you want them to catch on. You kind of want it to go viral. So this is the one I want to go viral, but it hasn't worked yet. So maybe this is the start of something good. So the way I think of it is that the cells are born with a genetic program, and they have, as I think of it, as a virtual lookup table. So they basically have a set of genes that are activated. And in fact, when we've done microarray analysis, and I think Paola has seen this as well, the interneurons actually have a surprisingly large number of receptors reflective of their mature state. I don't think they're activated, they're just RNAs. And I think shortly after migration, those receptors get, and here's the term I want everyone to use, weaponized. Yeah, that's cool, right? So what I mean is, they have this intrinsic program with all these receptors. They get there, and the receptors go into the surface, and the pathways become active. And these are like sniffers. So they get into that environment. Now they have all sorts of ways of responding to the environment and all these receptors to do so. But different environments provide different cues. And depending on which receptor you occupy, the cell says, oh, I must be in layer 4 of the visual cortex, and starts receiving appropriate afferents and making appropriate efferents in a way that's really specific. So some work by a guy named Robin Tremblay from Bernardo Rudi's lab has done this incredible job of mapping out 5A versus 5B basket cells. I mean, these are the nearest neighbors you can imagine. They're literally maybe 30 microns apart from each other. But they get totally different inputs. The VPA gets, the 5A gets input from the ventral anterior ventrolateral thalamus, where the 5B gets input. I may have this backwards. Maybe it's 5B. Michael, help me here. I believe, and the other one gets POM input, and they have, actually 5B gets VPVL input, and 5A gets POM input. And then their dendrites are completely different. So the ones in 5A are restricted to 5A. The ones in 5B actually skip over 5A and go up to layer 4. So these cells, which almost certainly have the same genetic program, by being placed in 2 microenvironments, do totally different things.
I got pretty much excited with your results regarding the changing cortical connectivity after altering thalamic activity. So the question again, where were the retrograde neurons after the cortical test? In which nuclei? Was it VPM or PO as well? And did you try to...
There's always more input to the neurogliaform cells from ventral posterior ventra anterior thalamus than there is from POM. And so preferentially, those are the ones we lose. The ones that remain are still from there, and you pretty much wholesale lose the POM. But to be honest, it's not, our analysis isn't thorough or quantitative enough to say whether it's preferentially one or the other. I'd say superficially it looks like they lose more POM than they lose
in case of pyramidal cells, you also showed that pyramidal...
It actually get an increase in the pyramidal cell innervation. So if you look at one cell and your question was how much input does this cell get? How many afferents does it get? That number doesn't change that much. What changes is the weighting of it. So whereas originally about at about postnatal day 6 or 7, a typical neurogliaform cell gets 2/3 of its input from thalamus and only 1/3 from the cortex. When we remove the NR1 channel from it and get rid of NMDA signaling, that population now seems to get about 2/3 of its input from cortex and maybe only 10% of it from the thalamus.
Okay, thanks.
Yeah, just a very quick. Hello. I just want to ask you if you can explain a bit more about your first point, which you criticized the classificationist about this. And I am not very clear because all during your talk, you said these specific cell markers are important, but at the beginning you said this is you have a radical position against this kind of classifications.
Yeah. You know, I guess contradiction is the height of intelligence, right? So, no, I'm making two sides of the argument. Certainly, different cell types have characteristic markers that you can see in them. So, you know, by and large, if you see parvalbumin, that's probably a basket cell. But as people who work in the hippocampus will tell you, there are a population called bistratified that have both somatostatin and parvalbumin. And a number of people have argued in the cortex, for instance, there actually are populations that at least at the RNA level have those two markers. If you look at CG-derived interneurons, that whole population has the ionic 5-HT3AR receptor, serotonin receptor, and that's kind of ubiquitous about those, right? So it's tempting to look at that kind of information and make pie charts and say, here's group 1, here's group 2, here's group 3, right? But I think that gives, that's kind of an artificial dichotomy. Because, you know, for instance, 5-HT3A, while being in CGE cells of, for instance, the the neurogliaform class, there turns out to be a 5-HT3A negative neurogliaform population in the hippocampus that's NOS positive that Chris McBain has looked at. So if you looked at the markers, you'd say, oh, it's no 5-HT3A, it's not a neurogliaform cell in the hippocampus, but you'd be wrong. It looks just like the neurogliaform cell, but it doesn't have that marker. So my point is, markers are useful. We use them not only as a simple means of classification, but as a way of genetically targeting cells with Cree driver lines. But it can be misleading because it's not like a marker is synonymous with a cell type. So I think as clearly happened in this talk, get sloppy and start associating markers as if they mean a cell type with the truth of what it is, which is a marker is one of the many genes that's turned on in a cell. And there's probably multiple ways of turning the same gene on. And so the presence of a marker in and of itself does not define a cell type, even though they're useful in terms of targeting and manipulating populations in a specific way. Is that clear?
Yes, but the physiology, the first slide about the phase locking, according different markers, they have found some phase locking relationship in CA1. Do you think this is a real, or this is also kind of first artificial?
I mean, it's very real. I mean, it's beautiful data. So if you happen to be looking at an axoaxonic cell in CA1, and you're looking at a theta rhythm, yeah, by and large, they will fire at the peak of theta. If you happen to be looking at a bistratified cell in the same area, yeah, by and large, they'll fire at the trough of theta. But if you look at the exact same cell in the cortex or the striatum based on the same markers we're talking about, and so talk about them as the cousins, so there's not truly a bistratified cell in the cortex, but there certainly is what we would call axoaxonic cells. no one's really looked at it, but I wouldn't bet that they necessarily fire at the peak of theta because they're in a totally different circuit. So if you're going to restrict yourself to a very small area of cortex and a specific cell type, yeah, absolute rules exist. But that's a product of all sorts of influences on the cell rather than the cell being a type, or at least that's my opinion.
Okay, thank you very much.